Hi guys, it's Professor Bittner, and I'm going to give you your final exam review for chapters 1 through 10 and excluding chapter 9. I'm going to go pretty quickly, so if you need to rewind and play it back more slowly, you can do that. Number 1 is list the categories of the hierarchy of biological organization in order. We're going to start with the smallest: atom, molecule, macromolecule, organelle, cell, tissue, organ system, organism, population, species, community, ecosystem, biosphere. Number 2, list the 3 domains of life and tell which contain members that are prokaryotic or eukaryotic, unicellular, multicellular, or both. So the archaeans, the archaea, those are going to be prokaryotic and unicellular. The bacteria are also prokaryotic and unicellular. And then the eukarya are eukaryotic, and some are unicellular and some are multicellular. So some of each. Number 3, state the main difference between prokaryotic and eukaryotic cells. And so I would say the nucleus, presence or absence of the nucleus. Prokaryotic cells don't have a nucleus. They do have DNA, but it's not surrounded by a nuclear envelope. Whereas eukaryotic cells have a nucleus. The DNA is enclosed. The other main difference is size. There's a big size difference between prokaryotic and eukaryotic cells, with the prokaryotic cells being much, much smaller than the eukaryotic cells for the most part. Number 4, list the basic structures that every cell has. Every cell has ribosomes. So ribosomes, cell membrane, plasma membrane, cytosol or cytoplasm, and DNA, some kind of chromosome. So that's the only thing we can say that all cells have in terms of structures. All cells do some type of cellular respiration in terms of metabolism. For each of the 4 macromolecule groups, be able to identify monomers by name or by molecule structure. I can do a rough drawing. You can look up more particular drawings if you like, but a rough drawing would be, for example, for the monosaccharides, this is a monosaccharide. So a hexagon. And so that would be glucose, for example, a monosaccharide. An amino acid, that's the one that has an amino group, what's called the alpha carbon in the center, and then a carboxyl group, and then a hydrogen here, and then some kind of other side chain or variable chain represented by R. All right. Lipids are a little more, a little bigger. So this would be like a triacylglycerol. This is a double bond O right here, by the way. And then this is just a hydrocarbon chain here. So this is called a triacylglycerol. So the zigzag is just a chain of carbons and hydrogens. And then these would all be hydrogens here. So triglyceride. Phospholipid, we draw like this. And then nucleotide. Let's do a nucleotide. A nucleotide consists of a 5-carbon sugar, either ribose or deoxyribose, a nitrogenous base, which can be adenine, thymine, guanine, cytosine, or uracil, depending. And then a phosphate group here. So phosphate group, nitrogenous base, and then a ribose or deoxyribose sugar. Pentose means it has 5 carbons. All right, starting with a ribosome attached to the rough ER, describe the series of events called protein secretion. So the ribosome is producing this protein, this polypeptide, by joining amino acids together. As the first amino acid, starting with methionine, starts peeking out of the growing polypeptide, there is what's called a signal recognition particle, which escorts the entire ribosome, mRNA, and newly growing protein to a pore in the rough endoplasmic reticulum. And it's also a docking site where the ribosome can attach. And then the protein continues to be made, but it's threaded through this pore in the rough ER. Once the synthesis of the protein is finished, the protein is released, but it's now located inside the rough ER. It folds, it goes through by vesicle to the Golgi. In the Golgi, it's modified and sorted and sent out to the plasma membrane by a new vesicle. When the vesicle fuses with the plasma membrane, the protein is pushed out of the cell. Number 7, given a gradient description, be able to predict the direction of movement of any particle via simple diffusion, facilitated diffusion, or active transport. The key is that if it's a facilitated diffusion or a simple diffusion, that everything is moving with the gradient. And if it's an active transport, then that would be the movement against the gradient. So typically you have some kind of diagram of the membrane. You have this, possibly this channel protein shown here. You have some gradient here. Let's say there's more solute here and less here. So by facilitated diffusion, the solute would go from its area of higher concentration to its area of lower. Simple diffusion would be just straight through the membrane. Only carbon dioxide and oxygen can do that. And then active transport would just be going the other way. If you had some solute on the side that has less of a concentration, but the cell wants to move it across to where there's more concentration, that takes energy because that's called active transport. Be able to predict the direction of osmosis in a given scenario or diagram. So whichever way the solute wants to go according to diffusion, then the water will go the other way. There is no active transport of water, and so you have to figure out the direction of diffusion of a solute, and water will go in the opposite direction. So an example of this would be, for example, what we call the beaker problems. So the beaker problems, we have a beaker solution. Inside the cell has some solute concentration. Outside the cell has some solute concentration. In this case, by diffusion, the solute would want to go into the cell, so water will want to come out. One thing, and the arrow means the water movement. The water inside the cell, 95% water, but only 90% water outside the cell. So water will go from where water is more highly concentrated on the 95% inside the cell to where it's less concentrated, the 90% area outside the cell. Know the structure and components of the plasma membrane. The main structure of any membrane is the phospholipid bilayer. So all these phospholipids and the tails, hydrophobic tails. And then you can have these proteins, transmembrane proteins, typically drawn like that. You can have a protein that has little hexagons attached to it. That would be a glycoprotein. Hexagons are not the greatest. And then cholesterol. Cholesterol has like 4 or 5 fused rings, but I'm just drawing it like a little black squiggle. So the cholesterol is completely in the hydrophobic portion of the membrane. All right, question 10. Explain the steps of the catalytic cycle of the enzyme. The first thing that happens is the substrate, which is what we call the reactant molecule, fits into a special niche in the enzyme, which is a protein usually, or can be an RNA. And then the active site is the exact shape to hold the exact correct substrate. And when that substrate enters the active site, then the enzyme actually hugs or folds or pushes down on that substrate. That's called induced fit. And at that point, it's called the enzyme substrate complex, the both of them connected together in that way. Then the reaction happens, which is bonds are made and bonds are broken, or both. And then the enzyme releases the product, and the active site is ready for another molecule of substrate to go again. Extremely high temperatures and high and low pH will denature an enzyme. They'll denature any protein, really. And denature means breaks all of the bonds that are holding together the three-dimensional shape of the protein. So it's straightening out the polypeptide chain. Without the shape, a protein can't do its function. Even if the polypeptide chain is still intact, if it's not folded, then it loses its function. The purpose of cellular respiration is to transfer energy from the bonds of organic compounds, which is a potential energy in covalent bonds, into the bonds of ATP, which is also potential energy, but in the bonds of ATP instead of an organic compound. So that's a transfer of energy. Photosynthesis is a transfer of energy from sunlight into the bonds of an organic molecule, such as glucose. Number 14, explain the purpose of the cell cycle for a multicellular organism. It's for growth of the organism and repair of tissues, not repair of cells. The cells are repairing the tissues. By making more cells, the tissue is repaired, and for maintenance of tissues. So growth of the organism, maintenance and repair of tissues. And then the products of the cell cycle, that would be identical cells or clones, genetically identical cells. Each cell entering the cell cycle will end the cell cycle with 2 cells just exactly like it. So it's clonal. All right, so that's a really fast review of the first 10 chapters that we went over and the main points.